Okay, then let's begin. I'm very delighted to welcome Professor Derk-Jan Dijk as our esteemed colloquium speaker. Uh, he is a world-renowned expert in sleep and circadian biology and currently serves as a director of the Surrey Sleep Research Center at the University of Surrey. His groundbreaking research has provided profound insight into the regulation of sleep, the role of the circadian rhythm in human physiology, and the broader implication of sleep health for cognitive and metabolic function. With over 250 published articles and numerous achievements, including the prestigious Distinguished Scientist Award from the Sleep Research Society, he work stands at the forefront of sleep sciences. In particular, his interdisciplinary approach, blending physiology, neuroscience, molecular biology, and digital medicine, has significantly influenced both basic and applied research, shaping current understanding and therapeutic strategy for sleep disorders. So today, we will talk about novel approaches and technology for the study of sleep and circadian rhythm in health and disease. Let's welcome Professor uh, Derk-Jan Dijk.
Thank you, Jay. That's a very kind introduction. It's great to be here. Yes, I'm going to talk about novel approaches. I'm also going to step back in time a little bit to give a little bit of the background.
Mhm.
But first of all, you know, uh, why are we interested in sleep regulation or the regulation of sleep duration in particular? And of course, one reason is that the epidemiology shows that short sleep duration is associated with a large number of adverse health outcomes. And currently, we are funded by the UK Dementia Research Institute, and actually, this is a study published a couple of years ago showing that's the Whitehall study that short sleep duration is predictive of a 30% increased dementia risk. And we know that for example, the UK or Great Britain population, 35% of the people sleep 6 hours or less. So how is sleep duration regulated? I want to pay tribute to Serge Dan. Let me see whether I can get this away. Hopefully, okay. Serge Dan, who was one of my PhD supervisors, he was a biologist. He was a pioneer in circadian rhythms, and he thought very carefully about processes underlying sleep regulation. And then he said, well, you need to distinguish between hourglass mechanisms and clock-like mechanisms. And the easiest way to understand this, if there is an hourglass and you prevent the behavior to occur, like for example, sleep deprivation, there will be a further increase in motivational tendency. Then you stop the deprivation and sleep will be shifted to another time. If it's a clock-like mechanism, you can take away the behavior, but actually the subsequent timing isn't affected. Of course, now we know that what is most likely that it's actually an interaction between these two processes. And this is based on work by Torbjörn Augestad, but the simulations are from Ans Kelden. And this is a simulation of that Torbjörn Augestad experiment. Here, people go to sleep after 16 hours of wakefulness, and then on another occasion after 20 hours of wakefulness, 24, 28, 32, etc. Now, if it was a simple hourglass model, you would predict that the longer you are awake and the later you go to sleep, the longer recovery sleep would be. Yeah, and you can see this, you go from 8 to 9.5. If it is an interaction between an hourglass and that circadian process, then you would actually expect that first we have 8 hours, then after 20 hours of wakefulness, what you see is only 6.3 hours, 6.2, 5.3, 4.3, and then all of a sudden, there is this jump, and you can explain this simply by an interaction. And that's, of course, in essence, the two process model of sleep regulation with some modifications, which I will come back to. Now, my task as a PhD student was to test the two process model of sleep regulation. And we worked a lot with Serge-Dan and Alex Borbe. And this, of course, is the two process model according to the 1984 paper, where we have the circadian process, the homeostatic process, we have a circadian pacemaker, of course, there is light input, and here we see the interaction. Yeah, but you can ask a number of questions. What is the separate contribution and of the clock and hourglass to sleep timing and sleep structure? What determines the phase relationship between the circadian and the homeostatic process? What determines the phase relationship of the sleep-wake cycle with the external world? I'm not going to go over all of these questions, but that was some of the things. Of course, working with Chuck Seisler in Boston, what we used was very much the forced desynchrony protocol to separate these two contributions. Now, this is a sleep-wake cycle of 24 hours, where you have day 1, whatever, through 7. Yeah, this is the minimum of the core body temperature rhythm. This is sleep. If you make a little matrix and you have here hours awake and hours of sleep, and here you have circadian phase, you will see that on the normal cycle, you only traverse this particular diagonal. Yeah, so you only have a very limited combination of C values and H values. If, however, you do a forced desynchrony and put people on, for example, a 28-hour day, and the sleep-wake cycle now desynchronizes from the circadian rhythm, you fill this entire matrix. And now you can look at the separate contribution. And that's what we did. Now, when you do it, there are all kinds of interesting observations. Well, first of all, if you look at the total sleep time as a function of circadian phase, there is this circadian variation, and that was well known since the 1980s. If you look at sleep latency, at the circadian modulation of sleep latency, now remember, these people are always 18 hours and 40 minutes awake before you put them to sleep at different circadian phases. Then you actually see that the longest sleep latency occurs at approximately 10 o'clock in the evening. Yeah, and that is associated and known as the wake maintenance zone. So the circadian drive for wakefulness is strongest in the evening and lowest in the morning. So maximum circadian wake propensity is in the evening, the minimum is in the morning, which means that if you draw the circadian thresholds of the C process, you need to have a skewness to the right. And that's very different from the 84 model and the 82 model, where the skewness was actually to the left or just centered on the middle of the day, but here it's to the right. So that was one thing, which I think is actually important. And that's wake maintenance zone is one of the most robust findings actually in the literature. We see that wonderful effect in the evening. So what you can do, and this is what we did back in the 1990s, keep people in the lab in Boston for a month, do the forced desynchrony, measure temperature, and then look at the propensity to be awake in those scheduled sleep periods as a function of circadian phase and time in sleep episode. Okay, and what you see this 3D plot, and this would be the trajectory we normally sleep on when we go to bed in phase with the circadian timing system. And you can see that here in the evening, when you go to sleep a little bit too early, you will hit the wake maintenance zone and you won't be able to fall asleep. But if you go to sleep at exactly the right circadian phase, you can stay asleep throughout the entire day. Now, of course, you know that when you go to sleep during the day, let me go back here.
Doc, what is the y-axis means here? Time in sleep episode?
Yeah, this is wakefulness within the 9 hours and 20 minutes
Oh, I
that
see.
people are in bed. So
Oh,
basically, it's the inverse of
uh-huh.
sleep efficiency.
What about the
So
time in sleep episode that
this
the
is from the lights out.
okay.
This is lights out until lights end.
오케이,
Yeah.
가차, 가차.
Yeah. Okay. So, and you can look this up. This is all published many years
Mm-hmm.
ago.
Mm-hmm.
Now, you can also, so that's the sleep side of things. It's important that you sleep at the right circadian phase. Shift workers don't sleep at the right circadian phase, so their sleep is disrupted.
Mm-hmm.
What about waking function? Yeah. This experiment we did here in Surrey, but Nayantara Santi, and she was interested actually in comparing men and women.
Mm-hmm.
So this is again a color coding, and here we see circadian phase, yeah, where 0 degrees is melatonin onset, and this is time awake,
Mm-hmm.
yeah. So when it's red, your performance is poor,
Oh, okay.
yeah, and this is in terms of accuracy.
Okay.
Now, when you are on the day shift, you are traversing on this white trajectory, yeah, and you stay in the blue, which basically means your performance, your accuracy is fine. If, however, you are a night shift worker and you work until 8 or 9 or 10 o'clock in the morning,
Mm-hmm.
but you start in the evening, initially your performance stays in the blue, but then in the morning hours, of course, your performance is impaired. And
Mm-hmm.
what you can see when you compare men and women, that in this sample, the impairment in the women was much more severe. Yeah. Now, then you can do some modeling. And again, this is also published quite a few years ago. And
Mm-hmm.
you compare the men and the women, the females, and you estimate the circadian amplitude. Yeah. Then the statistical analysis shows that women are more impaired during the night shift because their circadian amplitude is higher. Okay. Not because their homeostatic drive for sleep increases faster. So that shows that you can do funny things if you separate these two things. See, this is indicating what's the day shift and what's the night shift. And now you can also compare young and older people. This is again sleep. So this is time since start of sleep episode. This is circadian phase, and this is the corresponding time of day during entrainment. And here, warmer colors mean more disruption of sleep. And now you can see what happens if a young people goes to sleep at 11 p.m. That you stay in the blue. Even if the person goes to sleep at 2 or 3 o'clock in the morning, a young person still stays in the blue. On the other hand, if you go to an older person, the older person goes to sleep at maybe 11 p.m., that's fine. But as soon as the older person's sleep is slightly misaligned with the circadian clock, you see that at the end of sleep, sleep becomes very much disrupted. Yeah. And basically, let me see, actually, older people, older people would probably sleep better if they were to go to sleep even earlier, because they would be better aligned with their circadian phase. It also shows that older people's sleep is much more vulnerable to phase misalignment, so misalignment of sleep with the circadian system. Good. Now, so what I've been doing in the lab, we have this kind of modeling approach, and obviously Anne Skelden is doing a lot, where we know that there is both sunlight input, but also artificial light input into the circadian pacemaker. There are social constraints that obviously affect the timing of the sleep-wake cycle and thereby the homeostat. And then, of course, the sleep-wake cycle also has a big impact on light input into the pacemaker. Yeah, we are the only species that through our behavior can extend the photoperiod. So now we can ask questions, do experiments, see what the models say, and then ultimately we want to come up with some good advice. Okay. Now, what is important currently is to use novel approaches and technologies, yeah, to be able to record light, to assess the circadian pacemaker, to assess the homeostat in the community longitudinally. Yeah, and that's what you guys are very much interested in
Mm-hmm.
as well. And I think that's a very fruitful
Mm-hmm.
area.
Mm-hmm.
Now, as I said, we are also particularly interested in these novel technologies because currently we are focusing a lot on understanding sleep and circadian disturbances in dementia. Of course, especially in dementia, it is important that you have all kinds of technologies. So, why interest in sleep, circadian rhythms, and dementia? There is a bidirectional relationship. Undoubtedly, sleep and circadian disturbances are predictors of dementia, but dementia can, of course, also cause sleep disturbances. So, sleep and circadian disturbances are both a risk factor, a symptom, and a burden. But everybody is talking about it. But when you really critically read the literature, it's not so clear what the exact nature and extent of these sleep and circadian disturbances are. And then, of course, there are questions about mechanisms. And then, of course, ultimately, what kind of interventions can you come up with to improve sleep and circadian disturbances?
Mm-hmm.
And when you come to think about it, there are requirements to be able to do this research, and you want to have technology that is valid, acceptable, affordable, and scalable. Yeah. So that you can go to 24/7 monitoring. Actually, we want to go to 24/365 monitoring. Yeah, 24 hours, 365 days a year. And these are some examples of what we do. Now, you are all very well aware of all the novel technologies that are available,
Mm-hmm.
ecologically momentary assessment, blood-based biomarkers, um, data mathematical model fusion. We do a work on video, and then there are, of course, the nearables, the wearables, and the wearables. Yeah. And everybody is working on it. We have quite an interest in the contactless approaches because people with dementia often don't like to wear things. They don't like to have to recharge things, et cetera, et cetera. But I'll come back to that.
Mm-hmm.
So I'm going to go over a few studies. This is a study we published a few
Oh.
years ago
Mm-hmm.
now, which is an important study, I think, but
Mm-hmm.
it hasn't received all the attention
Mm-hmm.
it deserves. I think that's why I'm showing it
Mm-hmm.
here.
Mm-hmm.
So here we were really interested in the night-to-night variation in sleep and how this associates with the day-to-day variation in cognition, memory, and behavioral problems in Alzheimer's disease. Yeah, of course, you can ask questions about inter-individual
Mm-hmm.
type of association and intra-individual types of associations. And here we focused on the intra-individual aspects of it. So this was the design, approximately 2 weeks. We had, in this particular case, actigraphy, and then in the morning, people had a sleep diary. By phone, they were submitted to a cognitive test. And here
Mm-hmm.
you see some of the cognitive measures. And then we also had assessments of memory errors and mood. And also we asked the caregiver about their impression of the patient. Yeah. And so now you can do all kinds of interesting things. First of all, you collect all of these variables,
Mm-hmm.
but the question is, are these really all independent variables, or is there redundancy in the
Mm-hmm.
data set? Yeah. So one of the things you can do is basically, so for the cognition and mood variables, and here
Mm-hmm.
you have them: Karolinska,
Mm-hmm.
alertness, contentness, calmness, prospective memory, immediate recall, delayed recall, etc., etc. Yeah, and then you can simply look at how these variables and these are the deviations from the mean. Yeah, so this is actually the intra-individual variation, whether they are significantly associated. And you see that there are only very few red cells. Yeah, so which means the red cells are the measures that are significantly correlated with each other and to some extent redundant. But most of them are independent. So we kept the original mood, cognition, and etc. variables. But if you go to the sleep measures, where you have subjective time in bed, subjective total sleep time, subjective sleep latency, but you also have the actigraphy total sleep time, etc., you see that there are many red
Uh, related.
cells, which means that many of them are really dependent on each other. Yeah,
Mm-hmm.
and so what you then, of course, do is a principal component analysis. So we did a principal component analysis on the sleep variables, and then we came up with four factors. One is really related to sleep duration. The other is to sleep quality and sleep continuity. Yeah. And sleep quality, for example, is completely subjective. Yeah. Sleep continuity is to the larger extent based
Mm-hmm.
on objective measures. Yeah. So the next thing you can do is see how these principal components associate with aspects of performance. Now, so here we are looking at the inter-individual variation. Yeah. So this is, and what you can see is here we look at immediate recall, yeah, on the memory test. And here we have the principal component one, sleep duration. And we had three groups, the controls, the MCIs, and the ADs. And you see there is a negative correlation. Yeah. ADs, principal component one, sleep duration, which contains time in bed. They spend more time in bed, but their immediate recall is poorer, and the
Mm-hmm.
controls do the better. Yeah.
Mm-hmm.
So that's one observation. But now you can go to the intra-individual variation.
Oh.
So here we have the 14 days.
Mm-hmm.
Here we have everyday memory errors. Yeah. And you see the color codings for the Alzheimer's, the MCIs, and the normal controls, and of course the Alzheimer's have more errors, yeah,
Mm-hmm.
than the controls. And here you have the principal component one again, duration, and you can see that the Alzheimer's tend to sleep more, yeah. But you can also see that, of course, there is a lot of variation, yeah. And here this is the distribution of the deviations from the individual means. But what you next can do is look at the associations between sleep continuity, for example, and
Mm-hmm.
sleepiness, everyday memory errors, and behavioral problems. So this is in a linear mixed model. So you're really looking at the intra-individual variation. Yeah.
Mm-hmm.
And if you model this, and now we're looking at continuity of sleep, you see that if your sleep is more continuous, yeah, people report themselves as being less sleepy on the Karolinska sleepiness scale. Yeah, and you see this in all three groups. Yeah, if you look at everyday memory errors, you see that there is actually there is an interaction, but you see that in the ADs, more sleep continuity, better sleep continuity is associated with fewer everyday memory errors. Yeah. And this is the intra-individual variation. And you can also look at this is a revised memory and behavioral problem scale, which is completed by the carer, but we only did this in the Alzheimer's and in the MCIs. You see that if your continuity on the night before has been better, then the next day there are fewer behavioral problems. I
So
think
what's
that's
the
pretty.
definition of sleep continuity here?
Well, sleep continuity is based on this factor, on this principal component
I see.
analysis. Yeah,
Ah, I see.
and if you look at the weighting of the
Ah,
various variables, yeah,
uh-huh.
and so that the number of awakenings has
Ah,
a
it's small.
negative
I see.
weighting. Yeah, immobility
Mm,
immobility has a positive weighting.
I see.
Yeah, and the length of you see that, so this is really sleep continuity
Mm, Okay, I
based
see.
on these measures. Yeah,
Hmm.
that's pretty neat, don't you think so?
Yes, very good.
Um,
I see. Clear.
so but the principal component approach, I think, is very powerful, because then you really get to the essence of these aspects of sleep. Good, good. So now I'm going to talk a little bit about. We talked about biomarkers, yeah, and one of the things we were interested in is whether you could assess circadian phase by simply looking at the transcriptome in blood. We know that there are circadian oscillators everywhere in the body. We are interested in the circadian phase of the suprachiasmatic nucleus. We know that one core marker
Mhm,
is the melatonin
mhm,
rhythm, but to
mhm,
assess the melatonin rhythm, you need time series of samples. Yeah, and we really wanted to know whether we could just take one sample or two
mhm,
samples in
mhm,
blood and get a good estimate based on a biomarker panel uh, of the transcriptome that could get a good marker for melatonin phase. So we did this in a study, which is also published 10 years ago, um, in Surrey, where we did a forced desynchrony protocol. So first people were sleeping in phase with the melatonin rhythm. Then we shifted their sleep, and now they're sleeping. This is the melatonin rhythm as we measured, and then they were sleeping out of phase. So we collected time series of blood samples, so we could measure both melatonin, but also the blood transcriptome. You can see that the cortisol and the melatonin rhythm are not shifted. Yeah, this is because light was very low during the wake period. So this is cortisol and this is melatonin when people are sleeping in phase in
mhm,
blue or out of phase in red. But then we could also look at the transcriptome.
mhm.
So this is a heat map of when people are sleeping in phase. So they're
Mhm.
sleeping at the biological night.
Yes.
And then you see a large number of transcripts that are rhythmic. So
Mhm,
here we have plotted all the transcripts that are rhythmic. These are
mhm,
day peaking transcripts and these are night peaking transcripts.
mhm,
When you're sleeping out of phase,
mhm,
there is quite a disruption
mhm,
of the transcriptome. Yeah.
mhm,
So if you want to have a blood-based biomarker, that's a little bit of a challenge. Yeah. Can you still come up with a marker for melatonin based on the transcriptome, even though the temporal organization of the transcriptome in the periphery is so much disrupted? Yeah.
mhm,
And actually, when you look at all of the transcripts and you look at the rhythmicity, you can estimate the circadian contribution and the sleep contribution to the rhythmicity of all of the transcripts.
mhm,
And then there are transcripts that are completely circadian driven. Yeah.
mhm,
So this is a transcript. Uh, I guess this is the average of the transcripts where they have a nice rhythm when you're sleeping in phase. They
mhm,
also have a nice rhythm when they're sleeping out of phase.
mhm,
Yeah. Uh, but there are also transcripts where there is basically no circadian effect. So they are peaking during sleep when you're sleeping in phase, but they're also peaking uh, during sleep when you're sleeping out of phase of the melatonin rhythm. Okay, and you can do this for all 20,000 transcripts, and you can come up with these kind of plots, and this is all published. But then you can apply some simple machine learning approaches. In this particular case, partial least square regression, and then you can come up with a set of transcripts features, and you can then predict melatonin phase from that feature set. Yeah, and this is on the basis of two samples, I think. Yeah,
mhm,
and you see that there is a reasonable good correlation with melatonin phase. So that's one thing we have been doing. And again, this is published in eLife. Um, you can also look at the transcriptome now and say, is there a marker for how long you have been awake? Okay. And this was published back in 2013. There are transcripts that really go down with time awake. So this is doing a constant routine. There
Mhm.
are other transcripts that go up when with time awake. Yeah. And how many transcripts go up and down is affected by the prior sleep history.
Mhm.
So that if prior to the constant routine, you have only had 6 hours time in bed uh, versus 10 hours time in bed, actually, we see that many more genes or transcripts are now responsive to time awake. Anyway, so now you can ask the same question and do some kind of a machine learning approach and
Oh.
see whether you can get a biomarker for time awake. Yeah. And actually, we did manage to do this. We published this in Sleep a couple of years
Uh-huh.
ago.
Uh-huh.
You can come up with a set of transcripts
Oh.
that have an accuracy of 92%
Uh-huh.
in terms of estimating whether you haven't slept for 24 hours or more. Yeah. And these are some of, and here we use elastic net. So that's one of these regularized regression
Mhm.
approaches.
Mhm.
And it's that is doing very well. Anyway, so that's a kind of one aspect of the biomarker side of
Mhm.
things. Now, the important thing is this algorithm works on an individual, even if the algorithm has never seen a blood sample of that individual before. Yeah.
Mhm.
So that in principle is quite powerful. Good.
Mhm.
Now, as I said, we are interested in blood-based in biomarkers also for Alzheimer's disease,
Mhm.
and there is a lot of interest indeed,
Mhm.
and it is predicted that some of these blood-based biomarkers will be used for
Mhm.
diagnosis and monitoring disease progression. One of them is p-tau 217.
I see.
And one of the things we were interested in, just from a circadian perspective, how constant are these transcripts over the 24-hour day? So this is a study we did, and also this, it was published recently.
Mhm.
We had a
Oh.
group of MCIs, Alzheimer's, their
I see.
study partners, and cognitively intact older
Uh-huh.
people. If you simply look at p-tau 217, there is a clear separation between the groups. Yeah.
Mhm.
If you look carefully at the time course of p-tau 217 and you express the values as a deviation from the average value in that individual,
Mhm.
you see a very clear diurnal variation. Yeah.
See.
With lowest values in the morning and highest values
I
in the
see.
afternoon and evening.
Decreasing.
Now,
Oh.
the magnitude of this particular variation is approximately 15%,
I see.
and that's very similar to the 14.7 increase in p-tau 217 over 1 year in amyloid beta positive mild cognitive impaired patients. So 12 hours equals equals 12 months.
Oh.
That's I think that's quite interesting. And we did this for other markers as well, like amyloid beta 40, 42, the ratio, neurofilament light, and glial fibrillary acidic protein. So I think this is an important part, things to keep in mind when we talk about biomarkers for diagnosis. Okay, time of day matters. Of
Mm.
course, we don't really know what's driving this rhythmicity.
Mhm. Mhm.
It's diurnal, whether this
Mhm.
is a circadian rhythm or whether that's related to the sleep-wake cycle or posture or meals. But that's a question to be answered. Good.
Mhm.
So now I'm going to go and talk a little bit about some of the technologies we have recently tested. And the protocol we have been using, in the first instance, we took older people. Now, these weren't necessarily healthy people, but they were cognitively intact. They had all kinds of medical conditions, and we gave them the devices to take home. And then after 2 weeks of recording, excuse me, we brought them to the sleep clinic. Yeah. And then we could compare the devices against the gold standard. So, this is an example of one of the participants
Wow.
in one study with 45, 78, 70 years of age. These are some of the demographics. Now, so this is an example of 14 days of recording at home. And here we use the ActiWatch Spectrum, the Whittings sleep mat, and the Amfit, which is also a sleep mat. I'm not going to go through the data in great detail. They also completed a sleep diary. Yeah. And the sleep diary is indicated here by these triangles.
Yes.
And you can easily see, obviously, this is the nocturnal sleep period, and these devices certainly indicate well when a person is in bed. You can also see that there are daytime naps. If they are in bed, they are detected by these devices as well. But now you can do all kinds of simple analyses and evaluations. So one evaluation is if under the mattress Whitting sleep analyzer time in bed estimate, how well does this correlate with the sleep diary time in bed? And you can see that it correlates very well. Yeah. So that means that if you're interested longitudinally to know how much time does a person actually spend in bed, you don't need a sleep diary anymore. This device, you plug it into the mains, it never has to be recharged, and you can record data for months or years in a row. We estimated or we validated to what extent the device accurately detected when a person was out of bed by comparing the video PSG to the Whitting's out of bed estimates, and you can see there is a good correlation as well. So now you can also estimate how often a person gets out of bed and how long they are out of bed. And obviously, simple actigraphy doesn't really give you that information in an accurate way. So then, we did another comparison. So this is the ActiWatch Spectrum algorithm run without sleep diary information. And the question was, this is the time, the time of going to bed according to the ActiWatch Spectrum analyzer versus the sleep diary estimate. And you see this correlation isn't very good.
Yes.
Whereas for the Whittings and the Amfit, that correlation is much better. This has also been published in Sleep, and we provide many more. This is work by Kiran Ravindran in particular, and we perform many more analyses. So when you have all of this information, you can now kind of make a comparison heat map of your devices. Yeah, Somnify is a radar system. Yeah, and then these are the sleep measures you are interested in: total sleep time, sleep onset latency, light sleep, deep sleep, and you can see how well each of these devices estimate those particular variables. Yeah, and these variables are PSG variables, and here the units are the standardized absolute difference. So you can look this up, and now for your studies you can say, I'm particularly interested in this particular sleep variable, and maybe this device is the best choice if I'm interested in this particular sleep variable. Good. Now, the interesting thing about these devices is they also can record heart rate and breathing rate. Yeah. So here again, you have data over 2 weeks, and now the color coding is for heart rate or breathing rate. And if you look carefully, you can see that heart rate, of course, is higher at the beginning of the night compared to the end of the night. Yeah, because you can see this from the color coding, and breathing rate doesn't really vary that much. You now can do an average for every night, so you can look at the average variation in heart rate and breathing rate night to night. And actually, the Wittings also estimates the apnea hypopnea index. Yeah, and we kind of have validated this, and this is the heart rate as estimated from the PSG, for the
Uh-huh.
polysomnography.
Uh-huh.
And this is the heart rate as estimated, the average per night, averaged by the Wittings, and you can see there's a very good correlation. This particular participant had some kind of cardiovascular problem.
Mm-hmm.
So this is the Wittings, this is the MFIT. We have fewer participants there. You can do the same thing for the breathing rate, for the Wittings, the MFIT, and this is the Somnify radar system. And actually, these devices are pretty good at this. Some of these devices, like the Wittings, also detects snoring. Yeah, and there are no false positives in terms of, when the Wittings detect snoring, then the person is snoring. There are some false negatives. Yeah, and that's because the sensitivity of the microphone is probably not sufficient. Because if you look at the PSG detected snore intensity during a snore event which is not detected by the
Mm-hmm.
Wittings, you see that the intensity is
I
lower. Okay,
see.
so, but nevertheless, and then you can also look at the apnea hypopnea index as estimated by the Wittings. This is in the lab, and you see a good correlation, and this is now the the average with a confidence interval of the apnea hypopnea index identified over 14 days at home versus the apnea hypopnea index as identified by PSG during one night in the lab. Yeah, and you see there is a reasonable correlation. But now the interesting thing is that you can see there is this enormous night to night variation in the apnea hypopnea index as identified by the Wittings, and it's going to be of interest to us to see whether that variation varies associates with variation in daytime function, for example. Okay, good. So in terms of the contactless technologies and the potential for longitudinal monitoring, my kind of slogans are, nothing to wear, no recharge, no sleep diary, automatic data upload, integration with other data. Yeah. So I think there is quite a potential for these contactless approaches. They're not very good yet at detecting sleep stages, and I certainly don't want to claim that they are better than certain wearables. Absolutely not. But the burden associated with these technologies obviously is much lower. So that basically means that they may have applications in dementia, or they may have also applications in longitudinal recordings and other populations. Yeah, and you can easily see that if you're interested in mental health, or
Mm-hmm.
you are interested in experiments where you do sleep restriction, and you simply want to know when is a person in bed, it can be extremely useful to use some of these
Mm-hmm.
simple devices. So this is an example
Oh,
which we published, and I should emphasize that we are part of the UK Dementia Research Institute Care Research and Technology Centre at Imperial College London and the University of Surrey. Yeah, and there are many engineers, but there are also clinicians. The center is led by David Sharp. He's a neurologist, and he has data scientists like Ail Sorek. Yeah, and they have together with Syrian Borders partnership they have trials where they have people living with dementia in the community at home,
Mm.
and they have implemented uh, these under the mattress devices like the Wittings. Yeah, actually, they were implemented before we kind of evaluated them. But now that we know what kind of information coming out of these devices is reliable or not reliable,
Mm.
we can be much more confident about what we do with the data. And this is a recording, from the 1st of May 2019
Mhm.
until the 31st of January 2022. Yeah. And these are one, two, three, four, five examples. Yeah. And blue is time in bed. Yeah. And what you can see is that in this particular individual, not much is changing. There may be a little bit of an advance of sleep, and maybe here there is more daytime time in bed. Yeah, but if you look at this individual, you see that initially things aren't too bad, but then the person spends more and more time in bed. It's particularly clear in this individual, yeah, where initially things aren't that atypical, but then all of a sudden there is this advance of time in bed. Yeah. And there's a lot of time in bed, and there also seems to be some time in bed in the afternoon or certainly during the daytime. Yeah.
Mhm.
And as I said, this was published as a published abstract supplement of Conference Alzheimer's Dementia
Uh,
2022,
uh,
and you can find this figure online. And as I said, this is a wonderful collaboration between Surrey and Imperial and AO. Soric has done a lot of work in visualizing all of these data. Good. So it is a large team. This is one photo.
uh,
Not sure all of the people that have contributed are on this photo, but I hope I've mentioned them. We have other collaborations. All of the work I've shown you has been published, and some of it quite recently, others many more years ago. And that's basically all I wanted to say. Thank you
okay.
very much.
Wow. Thank you. Okay. Let me